So, um, hello everybody, and thank you very much for turning up to listen to us speak. We're going to talk to you about gathering in all the stuff that we like to measure, and we want to show you the importance of some of the smaller bones and artifacts that we collect and how those can be interpreted, and some of our techniques to measure the chemistry of human remains. So we understand that professional archaeologists have challenges associated with excavating human remains. There's the burial conditions, there's the preservation, and the small size of some of the bones. For example, one of the things that we're doing at the moment is using ear ossicles as a proxy for maternal diet in some of our studies, and they don't come much smaller than that. Softer tissues like hair, fingernails, and even preserved tendons can also be used in our analyses. So I'm sharing with you a page from a catalogue. This is an adolescent female from medieval Ireland that we're working on at the moment. And this is the catalogue that I was given along with the samples. And this is the second page. And you can see that this individual had carbon-14 dating, aDNA, strontium and oxygen isotopes from her enamel. We did dental incremental collagen. We've got bone collagen values from the carbon-14 dating. And we're investigating potential links between the stable isotope values and scurvy, rickets, and tuberculosis, which is what you osteologists will identify on the bones. So we've had to think about this sort of thing before, and the next 3 slides are taken from a publication we did with specialists from Molar. So it's in a book by Boydell and Brewer, which a lot of the universities have available online, so you don't have to necessarily go out and buy it or pick it up from a library. And we produce this to sort of show the value of the samples that we take from tooth and hair, and how even when things are being reburied, if we can take samples, we can go back to them and learn a lot about the people that we're working on. So for example, with tooth enamel, we can go through different procedures to look at migration, to look at life histories, to look at seasonality. Bone and dentine collagen allow us to investigate the diet, but also we can see changes in physiology during tooth formation, where nutritional stress can produce a particular marker, and so can chronic illness. So with dentine and bone, again, we can look at migration, life history, and seasonality. So teeth grow during childhood and adolescence, and if they're still growing at the time of death, we can have data about the final few months of life. But hair and fingernails grow throughout life and record more recent events. Sometimes our samples come from unexpected sources. For example, the hairs that an eagle-eyed pottery specialist spotted on the underside of a plate which had been placed on the abdomen of a woman buried in post-medieval London. They were very interesting hairs. So my claim to fame is I was a dentist, and then I started to collect teeth from dead people instead. And I'm particularly interested, as Rachel said, in what happens around birth. So this is just to show you the sort of things I'm looking for in teeth. So this is a diagram of how teeth will develop, deciduous teeth will develop. And you can see, looking at them, that they start to form before birth and they carry on growing to about the age of 3.5. That's the age growing. So if you wanted to look at the values in teeth for some kind of information that you wanted to get, so diet or physiology, if we wanted to look at what was happening at about a year of age, we could look at different parts of those teeth to get the same information. And if we wanted to look at what happens before birth, if we take the first 0.5 millimetre of any of those deciduous teeth, that tissue is formed in utero, so it gives us a window on the womb so we can see what the mother was eating. So what sort of profiles do we produce from these teeth? So the patterns that we can see in the incremental dentine can be matched to the age of formation, like we've just seen on the diagram. But generally, the flatter the pattern that we produce, the more stable the nutrition source and content is in that individual. So the top left-hand corner, you can see somebody who was living quite happily on potatoes before the Great Irish Famine. And so throughout their childhood, they had a fairly stable diet. If we get changes at a particular period in life, it can be linked, for example, to migrations from one place with one diet to another. So the larger plot there with the black lines, that's a female who was born and eating potatoes in Ireland, and then as a teenager she moved to London and adopted the more mixed London diet. So we can see migration just in her teeth. We can also see things like, for example, a child going from being a lay child into the monastery as a monastic member, because the diets of the lay people and the monastery occupants can be very different. We can also see things like changes in the diet, such as potatoes to maize during the Great Irish Famine. So the bottom left-hand corner, you can see a dramatic change in the blue line, and that's somebody who was eating potatoes, experienced the famine, could no longer access potatoes, but was given maize as a child in the workhouse as a supplementary food. But because it's got such a different carbon value, we get this dramatic shift in the plot. Also, we can see periods of stress. So again, looking at that bottom left-hand corner, we can see what we describe as opposing covariance. So the nitrogen goes up at a point where the carbon goes down. And this is a really good indicator of stress, where the body's recycling its own tissues. And we've seen it in other populations as well, and also where there's periods of chronic stress. So where there's no dietary explanation, we think we can we're quite able to spot nutritional and physiological changes. So we do like to try and develop our methods. We try to improve what we're doing. And a recent PhD student we had in Bradford, Mandy Curtis, worked on developing a method to microsample dentine. So she was trying, instead of cutting horizontal slices, which is what I normally do, she used a micromill to follow the incremental developmental lines of the dentine. So it means better temporal resolution, so we can get 3 times as much data from the same piece of tooth. And it also means less averaging of the signal for short-term changes, so it's not as flattened. So I'm going to show you a couple of her plots. So you can see there's the plots that were made from incremental 1mm slices, and then overlaid on them are the slices that Mandy got from using the same tooth but using micromilling. And can you see how much more detail there is in both of those plots? So where we suspect that something interesting is going on, it's worth spending the extra time and effort in getting the better temporal resolution. And you'll see another plot along the same lines later on in the talk. So I'm going to hand you over to Hannah now, who's going to tell you what to sample and why.
Okay, thank you, Julia. So I manage the light stable isotope facility here at Bradford. And I thought I'd talk a little bit about the types of questions that we get and the questions that you may have about sampling and what to sample. So yeah, what to sample, which elements are best for sampling, how many, and what about animal bone, the importance of a faunal baseline. I'm going to show you a couple of case studies from work that we've been doing at Bradford to kind of tease out some of these things. The question of which element is interesting, and it again, I think the main question, the main thing is, what is the question? What are you asking? And because bone and teeth are constantly turning over throughout life, we can target different parts within the skeleton, different tissues, even if we've got good preservation, to answer different types of questions. Now, Julia's talked about that very early life experience, but we can also look at bones to tell us a bit more about what's going on later in life. How many is, how long's a piece of string? And how much money have you got? But also, again, it is about questions. And I'm going to talk, give a couple of examples of population studies that we did, where, well, and okay, I don't want to give it away, so I'll come back to that. But then the question of the importance of looking not only at the humans, but also at the animals that were living on that site, and how important that is if you're going to do any kind of dietary reconstruction. So I'll move on. Oops. There we go. There we go. So tissue turnover. We know that teeth, so both that's the collagen and then the enamel on the outside, are forming from prenatal up until adulthood if we're looking at the third molars, but mostly what we're looking at is the child diet. And this tissue doesn't turn over, so you've got that trapped record of what was going on in those childhood years. The advantage of dentin is that it's also formed incrementally, so we can get a very well time related changes within diet. It becomes more tricky when we start to look at bone, and bone does remodel throughout life. And so by looking at targeting different tissues that turn over at different rates, we can try to target bone that has been laid over, slowly turned over bone that might give us information about earlier life or more of an average life value, versus faster turnover bone that's more likely to give you information about the diet of that individual within the last few years before death. Various researchers have attempted to find out which are which bones turn over the fastest, which bones to target. And you'll notice a lot of studies, certainly studies I've been involved in, where we are sampling, we're trying to look at changes within the life history of an individual. And we tend to take a tooth, a rib, mid-shaft femur, to try and get that kind of, to extend the life history information. Interestingly, just last night in preparation for this, I was reading a paper that just come out in the American Journal of Biological Anthropology, where a group have been looking at forensic samples and using bomb curve data and targeting different bony tissues. And what they've suggested is that actually, if you want to look at that kind of the older, so the slower turned over materials, something like an ulna might be the thing to go to rather than the traditional femur. They've also suggested looking at vertebrae, so trabecular from vertebrae instead of rib. Now, this is a small study and they were very old information. So I have, sorry, old individuals. So I have some caveat about that. I think there's more work done. But from the point of view of sampling, I would suggest if you want to get as much information as possible out of a skeleton would be to look for a tooth, a fast turning over material, so trabecular bone, be that from a rib or from vertebrae, and then a mid-shaft long bone or perhaps a bone from the skull to give you that collagen that was laid down in earlier life. Depending on preservation, of course, we can also look at hair and nails, and that's a lot of what we've been doing recently in the lab, working with modern individuals to try and tease out how the information we're getting in our carbon nitrogen is actually related to the diet that the people were eating, the physiological stress that they were under. And there you can get really good time resolutions, short time periods, be that weeks or months. I was just going to talk about a couple of case studies and how we've used isotopes to look at dietary reconstruction. So this first one was really my big introduction to looking at population-based studies with isotopes. And it was part of the NTRANS project. And here we were looking at Iron Age transitions across Europe, but specifically around Slovenia. And it was an interesting period to look at because, whoops, it's the wrong thing. Hang on, technology. Interesting period. The graves that had been excavated had a range of grave goods. There seemed to be a lot of stratification within the cemetery and also different types of burial practices. So the archaeologists came to me and said, oh, we want to know, were people eating different things and how might that relate to their grave goods? Are we seeing differences in diet due to social status? So I got the question of what to sample, how many, what animal bones, etc., and which kind of led me on this journey of thinking about these questions. So for this study, we took a rib sample, a tooth, and a mid-shaft femur sample from about 100 individuals. We also inhumation individuals and did carbon nitrogen isotope analysis on them. This is Rebecca Nichols, who was the PhD student working on this. And this is just some of the data that we got from some of these sites across Slovenia. And what we've got here is the coloured in ones are the rib samples in the diamonds. Then we've got the squares, which are the femur samples, and then the dentine samples, which are the circles. The dentine is up towards the top, and that kind of makes sense because we may be picking up breastfeeding signals and things that might elevate the nitrogen. But the main picture that we got from this is that it's pretty boring, really. This is just a small subset of what we looked at. We looked at all 100, but generally, all of these populations around Slovenia at this time had a largely homogeneous diet, omnivorous terrestrial diet, and low real evidence of marine protein. There was some input from millet, which was an interesting question for this time period in this region. But in general, similar protein across the sexes was not, we didn't see any difference between different grave goods. So they weren't eating a higher protein diet if you had a higher status grave goods, for example. So all a bit dull. Now, what was interesting was the first 10 samples that we did from adults could have probably encompassed what we got out of 100 adults in the end. So that question of how many to sample, it really depends on how much variability you have within the cemetery. But it also brought up something else that was interesting. And that is something that Julia and I both feel very strongly about, is don't ignore those outliers. So I've circled this bit here. So these are both from the same individual. We've got a dentine and a femur sample from this individual. And we wanted to look at why they were such an outlier from the rest. And so what do we know about this individual? First of all, it was a young individual, so it was aged at 6 to 9 months, based on dentition and long bone length. Also, this individual had widespread porous lesions, plaques of new bone on them. I'm not going to go into detail. That's a whole nother talk about what we named scurvy baby, but we did do incremental isotope data on that, and it told an interesting story in both the carbon and the nitrogen about what potentially was going on with this individual. So if you want to read more about it, it's down here. But I guess the take home in this is look at those outliers. They can usually often be the most interesting stories that you've got, and dig deeper into them. Okay, the second one I wanted to talk about, and partly this because I think potentially Holly may be on the call, who did lots of this work, but also it two cautionary tales that came from it. So this we've called the fishy diet population study. So this was a medieval settler cemetery in Patdom, which is in Romania, or was in the Transylvanian part of Romania. Some initial isotope analysis had been done by the archaeologists working on site, including Professor Bethard, and who'd done all the osteology on this site. And he noted that they had unusually high nitrogen isotopes within this population. They looked at a mix of adults and juveniles, and the juveniles, they suggested, well, maybe it's just a breastfeeding profile. That's what we're looking at. But also some of the adults were showing this high nitrogen. So the question was, why did these individuals have these unusually high nitrogen values? Possible causes for high nitrogen could be that they're eating fish, marine consumption. Now, if you look at where, I don't know if my little cursor's working here, where we are, we're not really near the sea. There are some rivers in that region, but it seemed unlikely. However, that was one cause for the high nitrogen. So there's freshwater resources, potentially weaning, as Julia can talk about in much more detail, and physiological stress. Interestingly, there were quite a few young children who died at this site. The demography was skewed towards young children. They didn't show typical signs of physiological stress. We didn't see the periostitis, enamel hyperplasia, stunting, etc. They looked, apart from being dead, reasonably healthy. And the other thing is, what about as animal husbandry practices? And we'll come back to that. So, I might jump through this. I'm assuming everybody kind of knows about this already. But when we're talking about nitrogen, the basic premises, I'm sure you've all heard of, you are what you eat, is that our bodily tissues, they turn over at different rates, and therefore they're trapping isotopic signatures from providing information about an individual's diet and surroundings at different times during their life. So the nitrogen of an organism is approximately 2 to 4 per mil higher than the food it consumes. Herbivores tend to be, there is variation here, about 5 per mil, and carnivores 2 to 4 per mil above that, so around 9 per mil, and omnivores sitting in between. So let's look at this data from PAP-DOM. And what we can see here is that it looks like a largely, in terms of carbon, it looks largely terrestrial, maybe some millet, but largely, yeah, no, certainly no indication of a fishy diet. The nitrogen in the adults as well, so now we looked at 95 adults, was up at about 11.5. We had some that were around 12 and 13. So yes, they was across the population, it looked like we were getting this high nitrogen. We also looked at sulfur. So here we've been developing carbon, nitrogen, sulfur combined isotope analysis on a single collagen sample, to working going from work that's been developed in Scotland on looking at smaller sample sizes. So looking at the sulfur, it looked very much like, again, terrestrial, no evidence of marine in that. We then also looked at some faunal, so these were terrestrial herbivores from this site. And if we look at the nitrogen, so again, the sulfur is looking very terrestrial. We look at the nitrogen and we can see it's up at about 7, so that's quite elevated. So our conclusions for this study were that no, it didn't seem to be that there was a lot of marine input in these individuals. Having looked at the fauna, it's more likely that we're looking at animal husbandry practices, potentially manuring, that meant that the people who are eating these animals, these herbivores and these omnivores, that's why they were getting the elevated nitrogen, which hadn't really been looked at before at that site. So what's new and emerging in our lab? A lot of it is a work in progress. So yes, come back to us in a year. Smaller and smaller sample sizes. Yes, I mentioned we've been working on collagen and keratin. So with collagen, we've got down to about 1.2 mgs of collagen. Now, the good thing about that is that it means we can do things like incremental dentin isotopes, and we can get a carbon, nitrogen, sulfur value from individual increments. We've also been looking at things like nail and sequential keratin samples. We've been working with modern hunter-gatherers and looking at changes through the season and different types of diet. By then, with keratin, we can get down to about 0.8 mgs. We've also relatively recently invested in looking at compound specific analysis. So we've got an LC, so liquid chromatography, isotope mass spectrometer, that allows us to look at individual amino acids. And our lab technician, who has some experience with that, have been helping to develop that. Our aim with this is to look at and really break down. So far, we've looked at a bulk collagen sample. That's telling you about all range of 20 amino acids that are in a collagen sample, but some of these amino acids come in different ways. Some of them are directly traced from our diet, some of them are recycled within our tissues. So what we want to look at is, especially working with modern individuals where we have a dietary history and health histories, to look at which of these amino acids are most closely reflecting diet, and also what that might be telling us about metabolic processes going on and how that might affect the stable isotopes. The last thing we have been looking at organic oxygen and hydrogen isotopes. Now, organic oxygen is used quite routinely in forensics, in wildlife forensics, but not a lot of it has been done in bone. It is fiddly, but the potential is great to be able to take a package of bone and get information, oxygen information, which may tell you about mobility and location within the same sample that we've also got dietary information. It is huge if we can get there. But again, that's very much work in progress. Here's back to Mandy. With Mandy, we've also, so as well as doing the microsampling approaches, we've been looking at how we can increase that temporal resolution in bone. So punching out osteons, looking at circumferential lamella, looking at different episodes of bone being laid down in a compact bone sample, and whether we can differentiate between isotopic ratios from bone that was laid down at different periods within an individual's life history. We've also been applying her method to archaeological samples. So here is a tantalizing plot. So this is for an individual from New Bun Hill Fields. This was part of Ruth O'Donoghue's PhD, working with Mandy. And here we can see the black lines are where we used Julia's approach, where we took the 1 millimeter sections, the dentine, and we've got smooth profiles. And then we've got the micromilling, and we've got a lot more variability going on. Now, if we look especially in the red lines where that nitrogen is, it's tantalizing because the green boxes are showing the approximate times in which enamel hyperplasia is formed in this individual. And they seem to match up reasonably well with when we're getting these peaks in nitrogen. So that is, watch this space, I think. We need to do more, but we're quite excited about that prospect of being able to look at these multiple renewed episodes of nutritional physiological stress. So just to summarize, I don't know if Julia wants to add something as well. Generally, so sampling methods can provide really high resolution records of dietary and metabolic changes right from before birth up until the time of death. Faunal baselines are hugely important if you want to do any type of dietary reconstruction. Context is also important when interpreting isotopes. The isotope data shouldn't be viewed as in isolation, but rather as part of an multidisciplinary approach, taking into account the osteology, the paleopathology, different biomolecular methods to look at the multiple causes that we get for stable isotope variability.
Yeah, and I think I'd just like to come back and say, when you are excavating in the field or collecting samples, just remember that some of those tiny samples might be the most interesting and most important. So look for those ossicles for us.